This research paper we chose to overview is Sara, Ajina, and Holly Bridges' Subcortical Pathways to Extrastriate Visual Cortex Underlying Residual Vision Following Bilateral Damage to V1. Before we get too much into the paper, we want to give a quick overview of some of the important terms and concepts which we'll be discussing later on. First and foremost, we want to briefly go over the primary visual cortex. This is what we call V1. V1 is the main input to the visual brain. When viewing a stimuli, the color, shape, and movement information from the thalamus are sent to different neurons within the V1 for further processing. Then, V1 sends these messages to different areas of the extrastriate visual cortex, which is a part of the occipital cortex. Going back to vocabulary now, we also have a new term called blind sight. This term can be called residual vision and refers to the phenomenon that occurs when participants have damage in their primary visual cortex. Here, patients are able to tell where an object is, although they claim not to be able to see it. On the other hand, when a patient's hemisphere is intact and has abnormal response to visual stimulation, we call this sight blind. We can think of these two terms, blind sight and sight blind, as being two opposites of each other. The last term we want to go over briefly is what we call HMT+, also known as the human motion area. fMRI studies identify HMT+ as an area which is activated by moving stimuli, even if the individual was reportedly blind. Moving forward, when it comes to damage to the V1, a patient can either experience unilateral or bilateral damage to their visual system. In patients who have unilateral damage to their V1, their intact hemisphere may also play a role in the residual function, or what we called blind sight. It is said that the intact hemisphere boosts perception. Many of these studies were done on patients who would undergo transcranial magnetic stimulation, or TMS, while completing a perception task. However, some studies have also shown the opposite effect and stated that the intact hemisphere has abnormal responses to the V1. However, when it comes to bilateral damage, not so much. While there is a large gap in the research, it is difficult to bridge this gap due to the rarity of the patients with bilateral damage. Studying bilateral damage to the V1 will provide researchers with the ability to understand whether pathways in the damaged hemisphere lead to residual vision, or if the intact side is required. To do this, the paper chose to re-examine the case of patient SBR. SBR is a patient who suffered bilateral damage to their V1 as a young adult due to a hypoxic event which left him cortically blind at the age of 22. Previous research on SBR studied his visual cortex at the age of 29 and compared it to that of healthy and excited individuals. As you can see here, he has bilateral lesions to his V1. This photo is displaying the field vision loss and lesions of other patients who have unilateral damage to their V1 in comparison to that of SBR's bilateral damage. The patients in the study have hemianopia, which is blindness in about half the visual field, usually on one side of the vertical midline. This can be seen here with the blacked out parts of the visual field. As you can see here, SBR is completely blind and is completely blacked out. Unlike the majority of research on SBR, this paper will study SBR's case in comparison to patients with unilateral damage to the V1, as well as healthy control patients. Due to the lack of research on bilateral damage to the V1, this paper seeks to determine SBR's pattern of response to visual stimulation in its HMT+ area and compare it to participants both with unilateral damage and healthy controls. The study will measure blood oxygenation level dependent signal, also known as BOLD signal, in HMT+ of both high and low contrast stimuli in both the ipsilateral and contralateral hemispheres. Finally, this paper suggests that by studying the microstructures between the lateral geniculate nucleus, LGN, and HMT+, as well as HMT+ in both hemispheres, that these tracts may be important when damage is bilateral.
In order to explore these comparisons, 2 study groups were recruited in addition to patient SBR, 8 patients with unilateral V1 damage and 8 healthy controls, with both groups averaging around 50 years old. Before the fMRI experiment, psychophysical testing was carried out for the patients with uni or bilateral damage to quantify their visual performance based on their ability to detect Gabor patches of differing contrasts. These patches are essentially striped circles, and the 5 contrast levels correspond to the degree to which they stand out from the uniform gray background they're presented on. Based on the participants' response of either perception or no perception during this 2 alternative forced choice task, the researchers were able to determine the presence or absence of blind sight before putting them in the scanner. To investigate any potential links between this blind sight and cortical activity in response to a specific contrast level, the percent bold signal change in the human motor area was calculated for low and high contrast stimuli in both the contra and ipsilateral hemispheres. Similar to the behavioral testing procedure described previously, each of the 5 different contrast levels were shown in the left and right hemifield in a position that matched the location of each individual schemata, that is, the area of the visual field with the most severe visual loss, as well as the equivalent position in the sighted visual field of the unilateral patients. To ensure subjects were not looking directly at the stimuli, they were required to stare at a fixation cross in the center of the screen and press a button when the cross changed from black to red.
Focus on the cross, and when it turns color, throw the ball. Okay? Nice.
Since patient SBR was unable to perceive the fixation cross, his eye movement was monitored by an experimenter and verbal instructions were given if his gaze shifted. All of the volumes gathered in a 3T scanner from these 10 fMRI blocks were pre-processed to correct for motion, reduce spatial distortion, and exclude all non-brain tissue. These functional images were then registered to the high-resolution anatomical scans gathered earlier in the study, as well as a standard MNI template, after which they were entered into a general linear model. To examine whether the increased percent bold signal change was related to brain anatomy, diffusion tractography was used to examine 2 pathways, the path between the lateral geniculate nuclei and the human motor area, and between the human motor area in the 2 hemispheres. This was done for patient SBR and both comparison groups in order to get a better understanding of how SBR's brain might be compensating for the bilateral V1 damage. Diving even deeper, diffusion tensor models were used to derive fractional anisotropy maps as indicators of the underlying white matter microstructure in 3 track profiles between the LGN and V1, the LGN and the HMT bilaterally. By coupling the fMRI data with diffusion-weighted imaging, Anya and Bridge concluded that bold activity does indeed reflect perception in SBR, as the response changed between low and high contrast stimuli, indicating recognition of the high contrast Gabor patch. Patterns seen in the diffusion tractography suggest this visual information is actually conveyed via a direct pathway between the HMT and LGN. Levels of residual vision and impaired integrity of the LGN-V1 pathway in patient SBR are equivalent to those seen in unilateral V1 damaged patients, suggesting that functional ability in one hemisphere is not required for blind sight. Setting SBR apart from the unilateral damaged patients, however, was an increased connectivity between the hemispheres of the HMT in absence of V1 input, meaning inter-hemispheric communication in the HMT may also be driving his blind sight abilities.
How does this study relate to course content? There's a multitude of ways the study relates to the material that we have covered throughout the course. In the beginning of the course, we covered bold responses and how it relays information about brain activity due to the ability to measure which areas of the brain are using oxygen in the blood, leading to distortions in the magnetic field that can be traced. In this study, the bold signal change in the HMT plus was measured for high and low contrast stimuli in the contra and ipsilateral hemispheres. This allowed the researchers to use the bold signal changes to detect activity changes in the given regions in response to manipulations.
The general linear model covered in the course was also used within the study, as each of 10 fMRI blocks described entered into GLM and contrasted against baseline fixation tasks to generate contrasted parameter estimates for each condition in every voxel.
This allowed parameter estimates to be extracted from ROI and the percentage signal change to be calculated. The regions focused on in the study would be considered regions of interest. There are multiple ways to conduct a study, such as being hypothesis or data-driven, and voxel-wise focused or ROI focused. This is a demonstration of how the region of interest analysis finds the right vicinity through anatomical location and functional location, and then extracts the data from experimental conditions to determine how activation occurs in this specific region. And that is why this is hypothesis-driven, region of interest focused. We have also learned to be wary of attentional confounds in class because fMRI data is highly susceptible to the amount of attention devoted to the task. This study addresses this by adding the aspect of the red and black cross in the fixation location while tracking their gaze to ensure attention is in the right place. This helped ensure activation is not simply due to an attentional confound. Limitations, conclusions drawn, and ways to extend. A limitation of the study was mainly the participants who were used. Due to the limited availability of patients that fit the necessary qualifications, there were only 8 patients used in the unilaterally damaged criteria. This leads to lower power in the experiment and therefore a less reliable outcome. Also, SBR is 29 years old, while other participants to which he was being compared were an average of 50 years old in the unilateral damage group and 53.2 years old in the control group. The conclusion of the study was that the patterns of residual vision in SBR are similar to those of patients with unilateral damage. Bold activity reflects perception in SBR, the bilaterally damaged subject. The response was greater when contrast was higher, suggesting that the bold signal change is related to visibility of stimulus similar to a healthy visual system. Also, there is interhemispheric communication in the presence of bilateral V1 damage. There were similar responses in SBR's contra and ipsilateral hemispheres distinct from healthy controls. This supports the thought that there is interhemispheric communication between HMT plus in spite of the V1 damage. The functional significance of this pathway and its influence on visual perception is still unclear and would need further exploration. Overall, the intact hemisphere is concluded to not be required for residual vision, but can be instead conveyed in the pathway between LGN and HMT plus. The study's importance lies in the understanding that those who have bilateral cortical blindness still have potential for rehabilitation. Without being able to look at the patterns of activation within the brain, those who had bilateral cortical blindness may have been ignored as candidates for visual training to improve residual vision. This study demonstrates that despite not having an intact hemisphere to boost function, there can still be patterns of residual vision, HMT plus activation to visual stimulation, and visual pathway microstructure.